Basics and genetics of color blindness. Color blindness is the inability to distinguish certain colors, usually caused by the absence or dysfunction of specific cone photoreceptors in the retina. The most common form is red-green color blindness, which is inherited as an X-linked recessive trait. Because males have only one X chromosome, possessing a single mutant gene causes the defect, affecting roughly 8% of males, compared to only 0.4% of females. Blue color blindness, or tritanopia, remains extremely rare. Trichromatic theory and classification. According to the Young-Helmholtz trichromatic theory, color perception relies on three types of retinal cones: S cones for blue, M cones for green, and L cones for red light. Defects in these cones are classified based on the nature of the impairment. Complete absence of a cone type results in dichromacy, indicated by the suffix anopia, such as protanopia, deuteranopia, or tritanopia. When a cone is present but functions with altered spectral sensitivity, it is termed an anomalous trichromacy, indicated by the suffix anomaly. Protanopia versus deuteranopia. While both present as difficulty distinguishing red and green hues, protanopia and deuteranopia have key physiological differences. In protanopia, there is a complete loss of L cones. Because no photoreceptors can sense long wavelengths, red light appears noticeably darker, and the visible spectrum is shortened. In deuteranopia, M cones are absent. However, because L cones still absorb long wavelengths, the visual spectrum remains normal in length, and green light appears misidentified in hue rather than dim. According to Ganong, deuteranomaly is the single most common form of color vision deficiency. Clinical assessment of color vision primarily relies on Ishihara pseudo-isochromatic plates, which screen for red-green deficiencies using colored dot patterns. For quantitative precision, Nagel's anomaloscope differentiates dichromates from anomalous trichromates, while Holmgren's wool test provides a simple matching evaluation. Accurate color vision is mandatory in safety-critical occupations such as aviation, transport, and pathology. Additionally, acquired color blindness can serve as a vital clinical indicator for optic nerve disorders, including optic neuritis and ethambutol toxicity.